you flunked the class. So remember that. But you shouldn't have to worry too much about it because, no, you will have to worry too much about it. I guess I said that the wrong ways. I'm slow, so you have a lot to worry about. So this is my second half marathon ever. I ran the first one last year in the Corvallis Half Marathon, and it's a wonderful race. So I can really recommend it highly. It's a lot of fun. So, but just 2 people in the class. Only 2 people in the class have to worry about flunking the class then. No, that's probably not true either, is it? Oh, okay. Walking on eggshells here. So it's Friday, you can tell, right? So, all right. So last time I got started talking about how we purify proteins, and I hope what you're seeing in this is we're using the properties of the proteins as strategies for isolating them. So that last one that I showed you, which was affinity chromatography, is a really, really powerful way to purify a protein if that protein binds to something that you know. You don't always have that advantage. Okay? And I should also point out that when I show you these methods, that the purification of a protein almost always requires more than one. It's rare you can just use one method. So there's no one way to purify a protein. All right? And so you have to sort of customize some of these techniques to purify a protein. Usually one will not suffice. All right? So that means you have to use your knowledge about protein structure. You have to use your knowledge of these techniques. Sometimes you have to try something and see if it works or not. Sometimes it doesn't work. So that's the sort of experimental part of biochemistry. And learning to do that and learning to troubleshoot those is really a very important part of learning to be a scientist. Okay. I didn't say a word last time about centrifugation. I just want to point that out to you. When we think about centrifugation, what we're doing is we're making a sort of an artificial gravity. Centrifugal force is generated by spinning a sample in a centrifuge rotor. When you hear about the G forces and so forth, those are basically experimental gravitational forces. And it's hard to find a biochemistry laboratory that doesn't have a centrifuge of some sort. And that's something that's really powerful because it allows you to separate on a very crude basis on the basis of size. Most of these techniques that I'm talking about have some relation to size. Centrifugation is really good for very big things. So for example, let's imagine I'm growing up a batch of cells. Okay? I'm growing a bunch of E. coli bacteria in a tube, and I would like to collect those E. coli cells. All right? Well, the simplest way for me to do it is to pop them into a centrifuge. They're big enough that the centrifugal force drives them to the bottom of the tube to make a little pellet, and then I can do whatever I want to with those cells. So for example, most commonly I would collect those cells, I would bust them open, and then get out of the inside of the cells those proteins that I'm after. So when I talked about this on Wednesday, I didn't really give you that, well, how do we get to the protein part? Anytime we're working with proteins, we basically are busting open cells and then taking that sort of soup that's left and doing that isolation of that soup that's there. Okay? So that's very simple terms. So centrifugation is really useful for us in terms of allowing us to separate very crudely on the basis of size. And the centrifuges that are used in a typical biochemistry laboratory can be pretty impressive. Okay? So a really powerful centrifuge known as an ultracentrifuge might spin samples at 100,000 times per minute. Okay? 100,000 RPM. That's pretty impressive. When you think about a car engine going 5,000 or 6,000 RPM, you think about that thing that's basically ballistic on your desktop going there, spinning things around. So you can generate some enormous centrifugal forces as a result of action of those. All right. Let's see. I think I talked about that, and I talked about that. So this one of the things I talked about last time was ion exchange chromatography, and I didn't click through those figures, but I think you get the idea about what's happening there. I want to turn our attention now to another separation technique that employs separation on the basis of size, and it does some cool things. So I'm going to first talk about agarose gel electrophoresis, and that's the most common type of electrophoresis that people do. How many people have run agarose gels? Yeah, okay, so you run those in high school now, so those are pretty commonly done. Agarose is a means of separation, which I'll describe to you in a second, It's used most commonly with DNA and RNA. We don't use agarose for proteins, and that's because DNA and RNA are much bigger than proteins, and we need some effective ways of separating things that are very large in comparison to proteins that are relatively small. Proteins are still pretty big, but in comparison to DNA and RNA, it's pretty large. Well, I like to start with agarose electrophoresis because it tells us some very cool tricks that we use in the separation process. Okay? So, first of all, what's agarose? So agarose is a polymer. It's a polymer that we actually derive from seaweed. Okay? Derive it from seaweed. Now, a polymer is a long chain. Okay? And we can imagine that that long chain, if we set it up properly, might make a mesh. So here's a chain up here. Here's a chain down here. I put some links in between them, and now I've got a mesh. And that mesh has holes in it. Right? Okay? So that's what agarose really is. So when I get agarose, I buy it from a supply store. I get a powder. That powder has all these various things in it. I can take that powder. I can mix it with some buffer, liquid buffer, boil it, and what will happen, just like Jell-O, if you've ever made Jell-O, what happens is that powder dissolves, and when it cools down, it solidifies, just like Jell-O does. And it gives something that's about the consistency of Jell-O. But in contrast to Jell-O, this has a very tiny mesh that's there, and those holes that I just described to you provide a separation of molecules. Agarose tends to have fairly large holes, and those holes are important because, remember, we're talking about separating fairly large molecules. Okay, so you got this pictured now. So I've got this gel. It's got the consistency of Jell-O, all right? And I basically pour it onto a slab. So I've got a slab of this stuff there, maybe about this thick, okay? I've got some little wells, and in those little wells, I put DNA, all right? How do I go through and do this process, all right? Oh, blast it. Don't I have that here? There's what I have. Okay. So this is what it looks like. This is called a gel apparatus. There's the thing I just described to you. I poured this, these bands I'll talk about in a second, but I poured this, and before I ran it, I had some little wells, which are just little places I can dump some liquid into there. I've got buffer covering this whole thing. Now I've got in my sample, I've got a variety of DNA molecules, and those DNA molecules are of different sizes. Now DNA is a polymer of nucleotides, and that polymer of nucleotides, every one has a phosphate. Phosphate nucleotide, phosphate nucleotide, phosphate nucleotide, okay? That's what that polymer looks like as I go across that. That means every time I say nucleotide, every time I have another base, I have another phosphate. The longer the chain is, the more phosphates I have. Right? If I were to think of the ratio of the size to the number of phosphates for any given DNA, it would be constant. Because longer makes more phosphates. So it's just proportional. The number of phosphates is just proportional to the size. Why is that important? Phosphate at buffer pH that we use for this is negatively charged. So now a DNA molecule, when I think about it, is really something that has negative, 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 right? It's what we call polyanionic. That becomes important because that's actually one of the important considerations in this separation. Right? So I've got my DNA sample. I put it in this little well right here. And it's got a mixture of different sizes, of different DNAs that are in there. I then take and I cover it all up, and I apply an electrical field to it. And that electrical field is set up so that I have the negative charge at this end, and I have the positive charge down at this end. Well, the DNA is negatively charged, right? The DNA is going to be repelled by this electrode. It's going to be attracted by this electrode, right? And so what's going to happen in the process of this is the DNAs are going to be pulled in the direction of the positive electrode. And it becomes a race. Who wins the race? Well, if we think about this, we've got these holes, these molecules are racing through. Which one's going to make it through fastest? The smallest or the biggest? The smallest. Okay? So the very first guys that make it through are the smallest ones, and they appear down here. And they actually appear as a band. A band corresponds to all the DNAs of the same size. They've all raced to the same point. The large guys, the big guys, have a harder time navigating through those holes. Okay? They go slower, and they're up here. So now I've just separated all these DNAs on the basis of size, and at any given point, each band, all the DNAs in that band are the same size. That's really useful. Okay? Well, how about if I want to purify proteins? What do I do? Well, I can't use an agarose gel because the holes are too big. The proteins just go shooting through it. We never see them. Okay? So we have to use something different to purify, to separate proteins. And for proteins, we have to use something called, actually I'll come back to that. We have to use something called acrylamide. So acrylamide is just a chemical. It has a different structure. And we can actually make this polymer within the confines of a gel by a chemical reaction. You don't need to worry about the structure or any of that sort of stuff. We have the same considerations we had for the DNA. I'm sorry, for the agarose, right? Agarose made these holes. What acrylamide is going to do is it's going to make holes, and they're going to be tinier holes. Okay? Acrylamide, when it polymerizes, when the gel sets, it makes a gel that has some of the same characteristics of an agarose gel, the main difference being it's just got smaller size holes to accommodate the smaller size, the protein molecules that are going to move through it. Now, if you've been thinking about this, all right, you're thinking, well, that's all fine and dandy. It's Friday. I know you're not thinking too much, but okay. At least I'm not. I better be or I'm in trouble, right? Okay. I've got these holes. That's all fine. But what's the problem with protein compared to DNA from what you guys have learned about proteins? I'll make you think about it here. Myriad of shapes, okay. They're what? They're not all going to have a negative charge. What happens if they have a positive charge? So on the first one, I had everything had a negative charge, and the amount of charge was proportional to the length. It was all beautiful. It was very simple. Proteins, some of them are positively charged, some of them are negatively charged, some of them are neutrally charged, some of them are very negatively charged, right? What's a biochemist to do? Well, all is not lost, okay? Biochemists, as I always like to say, are lazy people. And I can say that because I'm a biochemist. I'm a lazy person. So we figure out ways to make things happen that are easy. And early on, when people started working with proteins, they found a very cool trick that allowed them to give the protein a uniformly artificial negative charge. And there's a concept for you, right? Uniformly artificial negative charge. How do you do that? Well, it turns out that there's a detergent called sodium dodecyl sulfate, which you're more likely to call SDS. It's a detergent. If you think about detergents, detergents are soaps, and soaps are like fatty acids, one long nonpolar region and one end that has a charge. Well, SDS has a negative charge at that end. Detergents, as we've talked about before, just like heat, just like pH, will denature proteins, meaning they will unfold them. So a protein that starts out, as you're concerned, as a glob in a variety of shapes, when I mix it with SDS, it becomes linear, it lays out, and it gets coated uniformly with the SDS, the outside part of which is negatively charged. So for all intents and purposes, by adding SDS to the protein, what I've done is I've made this thing look like, okay, I've made it look like it's a negatively charged DNA. It's just got a long set of negatives. And the longer it is, the more negatives it has, just like the DNA had. So I've made this sort of artificial negatively charged protein, and now I have a mixture of proteins. I apply it to the top of my gel just like I did my DNA. And I set it up in exactly the same way so that the negative is at the top, the positive is at the bottom. So here I've applied my sample. I run the gel, so the negative's up here, the positive is down here. And exactly the same thing that happened with the DNA happens with the proteins. They go through on the basis of their size. The bigger ones take longer. The shorter ones move faster. And again, like I saw with DNA, a given band, all of the molecules in that band are the same size. Yes, sir?
So I know we're saying big and small, but what about shape of the protein? Does that affect anything?
Okay. So his question is, the shape of the protein affect anything? So I've made a simplification here. I said it unfolds it and makes it linear. Okay? But your question's a good one, because this is not a perfect technique. It's not perfect. So in some cases, we may not get it completely folded out the way that we want. So we sometimes see a little bit of an anomaly in the size of a protein based on what we'd expect, and it's probably due to the fact that it has a little bit of other shape associated with it. Okay? But it's a very good question. Yeah? Yeah, isn't that kind of a drawback? It's a very good question, and you're right on. All right? I want my protein purified. I don't want to have it in an unfolded form because I might want to get it out there and do something with it. For the most part, we don't use this technique to give us native, that is, functional proteins. We're usually characterizing the proteins with this technique more so than we are trying to purify. But it's a very, very good point. There are ways of purifying proteins and getting them out of here, but that's a little bit more involved than we can talk about here. But you're right on. Yeah?
Well, if you put the holes in the middle.
I'm sorry?
If you put the holes in the middle, positive guys go one way, negative guys go the other way, their charge just sits there.
Yeah, so it's a very good thought. So what he's describing is what we would call a native gel. Okay? You say, what if you put the holes in the middle, so the positives go one way, the negatives go the other way, and that will work, but there's a problem with that. Remember, we had to have uniform charge. So something that has a +1 charge will have a different force applied to it than something that has a +2 charge. If they're the same size, one's going to go twice as fast, but what if they're not the same size? I get too many variables. So yes, you can do that, but it's very difficult to interpret the results that you get as a result of the possibilities of charge and size. Does that make sense? Okay, so those are techniques of electrophoresis. Electrophoresis is pretty routinely done. It's usually done, as I noted, not so much to purify as much as it is to characterize. What are the size of the proteins I have in this sample? What are the size of the DNA molecules I have in this sample, etc. All right. Now, the last technique I want to talk about, and it's a little confusing, but it's one of the most remarkable techniques, I think, in all of biotechnology. Okay? So I've got to set it up for you. This technique actually uses two separation methods, one after the other. Okay? So I'm going to describe the first separation method to you, and the second separation method will be just like what you've already seen. So the first one you haven't seen yet. The first separation method, let's say I've got a batch of proteins. Okay? And they've got all different kinds of sizes, they've got all different kinds of charges. Right? The first method for separating, that I'm going to talk about here for separating these guys, separates them on the basis of their charge. It's kind of like what you were trying to get at here. Okay? Separate them on the basis of charge. Well, how do I do that? Okay? Well, I created something called an isoelectric focusing gel. Big mouthful of a name that's basically separating things on the basis of their charge. I'm going to tell you how it works, and this is the only confusing part of the process. All right? What I do is I take, you know those columns we talked about yesterday? We had all these little particles, all the little beads, right? I make another one. It's got a bunch of beads, but these beads have an interesting property. Some beads have on their exterior 100 negative charges. Some beads have on their exterior 50 negative charges. Some have 49, 48, 47. Some have 0 negative charges. Some have +1 charge. Some have +14. Some of them have +50. Some of them have +100. I have a complete spectrum of bead charges that go all the way from -100, in this case, it's an arbitrary number, to +100. Okay? Now I take this slurry and I just sort of shake it around. They're all mixed up. And I put my proteins in there with it. Okay? And then I apply an electrical current. Positive on one end, negative on the other end. What's going to happen? We're actually going to see a race. And the race is going to happen. The most negatively charged beads and the most negatively charged proteins are going to go to a certain place and stop. The most negatively charged one will go all the way to the end. The second most negatively charged one is going to get squeezed out by the most negatively charged one, right? So we can imagine that in this tube, when I'm done, I have everything from minus 100, minus 99, plus 99, minus 98, plus 98, etc., all the way down to the very middle where I've got 0. My proteins are aligned in exactly the same fashion because they get squeezed out by the same things. So proteins that have a minus 100 charge are way on one end. Proteins that have a plus 100 charge are way on the other end, and everybody else is in between. Size didn't matter. I don't have any holes. I simply separate it on the basis of their charge. Well, that's what this is showing you, okay? All right? So we have these things separated. It's calling pH, but for our purposes, we'll just call it charge from one side to the other. All right? Now, on one side, I have molecules that are the most negative. On the other side, I have molecules that are the most positive. I take that slurry, and it forms a sort of a gel is what it does, and I lay it on top of the gel that I separate proteins with. By the way, that gel is called a Polyacrylamide Gel. So we call the technique Polyacrylamide Gel Electrophoresis whenever we're separating proteins. It's called PAGE, P-A-G-E. And more importantly, it's got SDS in it, so we call it SDS PAGE. So this is an SDS PAGE gel, just like we did before. But now, laid across the top of it, I've got proteins from minus 100 over here to plus 100. They've already been separated on the basis of size. Now we're going to apply a negative electrode, and we're going to apply SDS to them, so now they all get uniformly negatively charged, even if they weren't before. And we run. So on this next time we run it, they separate on the basis of their size. We've just done 2 dimensions of separation. It's called 2D gel electrophoresis. Now you'll see the power of this in just a second. Up here on the upper left, I'm going to have high molecular weight, most negatively charged. Over here, I'm going to have high molecular weight, most positively charged. Down here, low molecular weight, most positive, etc. You can figure out what's going on in there, right? Guys in the middle are going to be pretty neutrally charged. Also separated on the basis of charge. I'm sorry, on the basis of size. Now, let's think about this for a second. This technique it's part of a discipline called proteomics. Proteomics allows us to separate all of the proteins of a cell and get a unique spot for each one based on this technique. So a typical cell might have 5,000 or 6,000 proteins. Okay? It's going to have some that are high molecular weight and negative, middle molecular weight and no charge, etc. But every protein is going to have a specific place it's going to migrate to when I do this analysis. So what? Well, when I'm done with this, I might get 6,000 spots. Some spots are going to be really dark, meaning I've got a lot of that protein. Some spots are going to be really light. Right? So what? All right? Well, let's imagine that I'm studying a cancer. Let's imagine that I'm interested in understanding the proteins of that cancer. Let's say I've got a liver cell. My grandmother's got liver cancer, and they extract a tumor from her, and I've got these cancer cells that are here. I want to know what proteins are screwed up in this cancer cell. Okay? Well, how do I know which ones are screwed up if I don't know what the normal ones look like? Right? So at the time they take out the tumor, they also take out a little piece of tissue of her normal liver. I bust open the proteins, and I do 2 gels. Exactly like this. All right? So I run 2 gels. One gel is my normal. One gel is my cancer. And I ask a very simple question. Where are the spots different? Where are the spots different? The cancer cell has certain spots the other one doesn't have. These are proteins that are being made only in the cancer cell, not in the normal cell. Maybe there's some spots that are in the normal cell extract, but not in the cancer cell. The cancer cell has shut off the production of those proteins. Maybe there's more in one than there is in the other, etc. We can imagine a whole spectrum of these things. But the beauty of this is not only can I look at this and see the difference in those spots, but I can also easily determine which specific protein corresponds to which spot. Was that your question?
No, I'm going to say, is it analyzed by a human or by a computer?
Good question. Let me come back to that. I will address that, okay? So I can easily determine what spot corresponds to what protein and the differences between them. Great for cancer. What if I'm testing a new drug? Does this drug negatively affect liver cells? Pretty simple. I can take one mouse, for example, give it the drug, take another mouse, not give it the drug, compare the two. Does this environmental pollutant cause skin cells to do something different? Bang, bang, right? And I can answer across all of the proteins very easily, is there a difference between the two? That's really an incredibly powerful technique. 2D gel electrophoresis has really transformed the way that we look at proteins in cells. Now, her question here was, do we analyze these by human beings or do we analyze these by computers? And we do a sort of a combination of the two. Okay? The best analyses and the most quantitative analyses are done by the computers. So the computer can actually take the output of that plot and determine how dense is that spot and compare to that identical spot over here, and you get a numerical listing of those two. Human beings, being human beings, tend to look at things as well. So humans will do that, but the best analyses are done by computer. Now, it gets even better than that in that robots actually do some of the analysis as well. So I told you that we can determine easily what the composition of each protein is in each spot, right? The way that's done is by analyzing the mass of each one. So we have to analyze the mass of the protein in each spot. And that turns out to be fairly trivial to do. A robot can be taken to actually cut out, recognize and cut out using some little tiny scissors and tweezers each spot, extract the protein out of it, and run it through a machine called a mass spectrometer. You probably learned a little bit about that in basic chemistry. The mass spectrometer says, here's the mass, bang. Here's the composition of amino acids in it, bang. Okay? Here's the identity of your protein, bang. Okay? That analysis, which is nothing short of magical in my opinion, can tell a scientist in one day the composition of 6,000 proteins on a gel. No, it's not like HPLC. That's a different technique. Yeah. Okay? Okay. So it's easy for us to tell, okay, well, you've got too much DNA polymerase in this cell. You've got too much of this ribosomal protein E13, all right? And maybe that's related to why this cell is growing uncontrollably. Okay? You've got too much of this oncogene, and yeah, oncogenes cause cancer. Bang, we've identified the problem. It's really, really cool, powerful technique. So let me stop there for a second and take any questions you might have. As you might guess, I think 2D gel electrophoresis is pretty cool. Yeah?
Why you said that the beads were negatively charged?
Yeah.
And you had, and they were specific to another negatively charged DNA or whatever.
Yeah.
Why do they not repel each other as
They do. They do. The most negative one makes the farthest, and it's pushing away the next most negative one.
So they don't come together and then migrate? They all migrate?
They all migrate according to how much charge they have. So the ones that get pulled the hardest are those that have the most charge. And on the other end, the ones that get pulled the hardest the other way have the most charge the other way. So there is a repulsion, and that repulsion results in the most negative ones and the most positive ones reaching the farthest ends.
But I guess what I'm asking is the bead.
Yeah.
You talked about that like was an attraction between them.
It is.
Right?
It is.
Why do two negatives attract each other?
They don't attract each other. They're attracted to the positive electrode. Remember, you've got a positive electrode. That's where the most negative ones are going. So the one that's got 100 negative charges has a real strong attraction, right? The one that has 99 charges is attracted, but it can't squeeze out that 100, which has a greater attraction, right? So I see your confusion, but you're forgetting the external forces that are attracting that. That's what's causing that gradient. It's the external forces, the positive at this end, the negative at this end. Make sense? Yeah? Well, the beads are sort of there to provide that squeezing out thing. So they allow the proteins to separate on the base. If we just put the proteins in there, all the negatives would be at one end, and all the positive ones would be at the other end, and there wouldn't be that gradient that we would see. That's why the beads are there.
They're kind of resistant.
They are a sort of a resistance, that's right. Yeah?
So is this done in 2 individual steps? So first you
It is 2 individual steps, that's right. And there is a sort of an art to this. That is, making this and applying this to the top is sort of an art. Yeah.
So, but once you wash them with the detergent, how does that affect the mass spectrometer reading?
It's a good question. So once you have the detergent on there, does that affect the mass spectrometer reading? It turns out that going through the mass spectrometer, the detergent peels off readily. So any individual molecules that are not covalently bound will be stripped away. Yeah?
So the proteins are actually stuck to the balls, right?
They're not stuck to it, no. They're just sort of surrounded by these charges as such.
So then it kind of goes like ball, protein, ball, protein, ball.
Yeah. Yep.
Is it expensive?
Is it expensive? It's not that bad. The biggest expense of it is probably the time investment, and that's true of many things in science. What it costs to pay people to do it. That's why you see robots being employed in some cases. What it costs to pay people to do these probably is the biggest expense in them. So I wouldn't say no. It's not prohibitively expensive.
Is it being done here?
Yeah, it's being done here at OSU. Yeah. Yep. We have some real state-of-the-art researchers at OSU doing mass spectrometry. Really phenomenal things that they're working on techniques for determining mass. Those are really powerful tools. Okay, so that's what's up with that. Now you asked about HPLC. Let me just briefly mention HPLC. It's not too difficult to understand. HPLC stands for High Performance Liquid Chromatography. HPLC, High Performance Liquid Chromatography. Some people mistakenly call it high pressure liquid chromatography because it actually does use and require high pressure, but the P technically stands for performance, right? How does this guy work? Well, it also works with columns. We talked about columns. We talked about beads, right? This guy has a little bit different strategy. Okay? So the beads for this are microscopic. The other ones I said might be a millimeter or so. We could actually see them with our naked eye. These guys are microscopic. And now, instead of having charges on them, or instead of having specific molecules like ATP on them, or instead of having holes through them, these beads have nonpolar tails sticking off of them. Long nonpolar tails. So each bead basically is a fairly nonpolar entity. This technique of HPLC that I'm getting ready to describe to you is called reverse phase chromatography. You don't need to worry about why it's called that. That's just what it's called. Okay? Reverse phase. Now, the reason we need high pressure is because these beads are so tiny, they fit together very tightly. You get millions of them in a given column of these. And so if you're trying to get molecules to move through there like we did before, instead of letting solvent flow through them, which is what we were doing with the other columns, we actually have to force it through with a pump. That's where the high pressure comes in. I put my sample at one end, and the materials in that sample start interacting with the nonpolar group sticking off. Let's think about protein molecules. Some proteins that are positively charged, do you suppose they're going to interact much with the beads? No. Negatively charged? No. Uncharged? Yeah! Sorry. All right? So this technique separates on the basis of the polarity of a molecule. The most polar come out first, and the least polar come out last. And in between are those that are all in between. So now we've got several ways of separating molecules. We've separated things on the basis of charge. We've separated them on the basis of size. We've separated them on the basis of affinity. We've separated them on the basis of overall charge with the isoelectric focusing. And now we're separating them on the basis of their polarity. You start seeing that, wow, you've got an awful lot of tools that you can separate things with So that's why, you know, you've got a lot of ways of separating proteins. There's no one way of doing it. Yes, sir?
Is that actually how they come out, the peaks or how much?
So, yeah, thank you for pointing this out. So what we see is a typical plot that would come out. These are for individual amino acids, but we could also do it for peptides, for example. Okay? This is what you will see, and one of the reasons people really like HPLC and it's such a powerful technique is because HPLC is very, very reproducible. You get very sharp peaks. Okay? And in this case, for the separation of amino acids, I see aspartic acid coming out very quickly. All right? And aspartic acid will always come out at this point, depending on the column that I'm using. So there's great reliability that's there. Now, this particular column is a little misleading because here's lysine, and you say, well, that's very positively charged, it's coming out last. They've done a little chemical manipulation here that we haven't talked about. All right? But the beauty of HPLC is a very, very reproducible separation, and you can separate molecules with HPLC that you can't separate by almost any other technique. It's really, really reliable. HPLC is used an awful lot in quantitative analysis of characterization of things. Okay. All right. So we're moving our way through these. We've finished talking about what I want to talk about now with respect to the separation techniques. I want to spend just a little bit of time talking about digesting proteins. And I'm not just talking about digesting them in your stomach, but digesting them for analysis in the laboratory. Okay? Not surprisingly, we use some of the enzymes that our stomach uses to digest proteins in the laboratory. Okay? I'll show you a couple of them. All right? Here's a polypeptide chain right here. Polypeptide chain. All right? And one end, we've got aspartic acid. There's the sequence of amino acids that are there. Okay? Most proteins, when we try to analyze them, are too big for us to handle. So if we break them into pieces, the pieces are easier to characterize, and then we can understand the whole protein by putting the pieces together. So first we have to get the pieces, and the pieces are fairly easily made by breaking them down using specific enzymes that cut at specific places. That's what these digestive enzymes do. These enzymes that break proteins down are called proteases. Proteases. Proteases work, at least most of them work, by recognizing a specific amino acid and cutting next to it. So trypsin, for example, the one that you see on the screen, you'll see more about trypsin later. It's involved in blood clotting. It's involved in digestive processes. Okay? It has some really cool things. Trypsin cuts right next to an arginine or a lysine. So it cuts. Here's the starting material, treated with trypsin, arginine, lysine, arginine, and we've cut this guy into pieces. And we've cut them into specific size pieces because they've only cut at arginines or lysines. Okay? We'll talk later about another protein, another protease called chymotrypsin. It's related to it. And I'm not going to give you the specificities here. That is, when I say where it cuts, that's called a specificity. Okay? I'm not going to give you the specificity because then you'll just think you have to memorize it. All right? I will tell you about a couple things that we use that you should know the specificities of. And yes, you should know it for trypsin as well. It's useful to know that. Trypsin is used very, very widely. Okay? Sometimes we break proteins down by something called carboxypeptidase. This is the easiest one. Okay? Carboxypeptidase starts at the carboxyl end, and it sequentially chews in. Doesn't have any specificity. Doink, 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 doink. Okay? So if I want to start breaking things down at the carboxyl end, I use carboxypeptidase. A third thing that we use in the laboratory commonly to break down proteins is a chemical. Okay? We use chemicals sometimes to specifically break down a compound. All right? And those are also very useful. So it doesn't have to always be an enzyme to break down a protein. The chemical that we use is called cyanogen bromide. And cyanogen bromide is one of the easiest ones to remember. It breaks right next to methionines. Wherever there's a methionine, it causes a break to occur in the overall structure. So there's a methionine right there. We see, and you don't need to worry about what the chemistry is or any of that, but the result is that a break will happen where there's a methionine. Gesundheit. Okay, so these reagents are really powerful, really useful for us to be able to understand protein and characterize protein structure a little better. Okay, questions on that before? I'm going to say a little bit about enzymes today, and then we'll finish for the week. Any questions about this? Yeah? It breaks it right next to methionines. Yes, ma'am?
The carboxypeptidase breaks at every amino acid?
Yes. So it starts at the carboxy end, and it just starts chewing inwards, kind of like Pac-Man. You guys are too young to know what Pac-Man is, so. I'm an old geezer. You already knew that, though, so. Other questions? Okay, let's introduce the topic of enzymes. We're not going to get very far on enzymes, but let's think about what enzymes do, all right? And we'll actually do it a little geeky here, too. All right? Now, enzymes, as I've said before in class, are remarkable things. They are biological catalysts. They're protein catalysts. They're proteins. They are almost infinitely better than chemical catalysts. I gave the example about a chemical catalyst like platinum that might speed up a reaction 1,000-fold, and some enzymes that are capable of speeding up reactions 100 quintillion-fold, 100,000 trillion times. Okay? Remarkable. So one of the things we want to do is we want to understand how enzymes do what they do. Now, it's important to recognize that what enzymes do, and this is going to surprise you, is they will not make a reaction go that would not otherwise go. All they will do is speed it up. They will make it react faster. They will make it get to equilibrium faster, but they will not make a reaction go that will not otherwise go. So enzymes, even though it seems like they're doing the impossible, really can't do the impossible. So we want to understand how it is that an enzyme does what it does. All right? So I'll give you one geeky thing today, and then we'll do a song. How's that? Okay? All right. The geeky thing. You learn in chemistry that reactions proceed as a result of changes in energy. Everybody know what activation energy is? Have you guys heard this in chemistry classes? Activation energy is energy necessary to get a reaction started. If we think about a molecule over here and a molecule over here, they bounce into each other with a certain energy. If they have the right energy and the right orientation, they will react. If they have the wrong orientation or the wrong energy, they're not going to react. They bounce off. Okay? That's the activation energy that's right there. All right? An uncatalyzed reaction has a certain activation energy to get up to what we call the transition state. And from there, it's downhill. It can go downhill forwards, or it can go downhill backwards, back to where we started. So it may not make it. It may make it to the next thing. Here's the energy of our reactants, and here is the energy of our products. And you can see that the energy of our products is lower. Energy has been given off in the process. This is an energetically favorable reaction. But we've got this barrier. We've got to get up over this hump before we can make this guy happen. That barrier is literally a barrier. It takes a lot of time for molecules to coincidentally hit each other in the right orientation and make this process go. Well, how do enzymes do it? Enzymes do it by lowering the activation energy. Okay? Notice, the reactants start with the same energy as they did up there. The products finish with the same energy that they did up there. The only difference was, how did they get there? Gesundheit. Now, I'd like to give you an analogy with this. The analogy I always like to give is, imagine, if you will, that we're sitting here in Corvallis, Oregon, and we're about 200 feet above sea level. Right? In theory, I could take a giant steel ball bearing and walk outside of the LPSC here and push it in the direction of the ocean, and the ocean being 200 feet lower than Corvallis, if there weren't trees blocking the path, it should make it there, right? No, it's not going to make it there. The reason it's not going to make it there is because there are energy barriers in between here and there, right? So you say, okay, I'm very smart. I'm going to get the class of BB350, and we're all going to grab this, get behind this giant steel ball bearing, and Kevin's going to give us extra credit if we do this, and we're going to push it to the top of Mary's Peak, because that's the tallest peak in the mountain range here. And if I have no trees blocking us, we're going to push it off, and it's going to go doing, 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 and it's finally going to make it down to the ocean. And that's another strategy, and that'll work. Unfortunately, that's this strategy. Go to the highest possible place, you know it's going to make it there. The enzyme says, well, hey guys, we got mountain passes. All we really have to do is get to the highest mountain pass. It's going to be way lower than Mary's Peak is going to be. It's going to be much easier to get to the mountain pass, and once we get to the mountain pass, gadoing, 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 and it's going to make it down to the ocean. That's what the enzyme's doing. It's finding the mountain passes. Okay? It's finding those mountain passes. Yes, sir?
So the same amount of energy is released, though.
That's right.
Even catalyzed, and it takes less energy to start.
That's right. So it takes less energy to get the reaction started, but the same amount of energy is released in the end. That's right. Other questions? You ready for a song? Yeah. Okay. You got to sing loud. You guys did a pretty good job last time. Let's hear you. This one is to the tune of an old Woody Guthrie song, which you may not remember, but you probably have heard before, called This Song's for BB350. So, it's written for a different class. You see it's at 1 o'clock, so it's not 1 o'clock, but it goes as follows. It's 1 o'clock and Ahern's talking, Henderson and Hasselbach and PKAs and buffers I should know. This song's for BB350. I hope that maybe he'll think the way we wrote our answers wasn't crazy. I really need the partial credit, so this song's for BB350. It's really groovy that it improves me watching lectures and QuickTime movies. I really need to go and download those podcasts for BB350. I'm feeling manic, I'm in a panic, I'd better study my old organic. It has reactions that I need to know. This song's for BB350. I know he said it, that's why I dread it, 'cause I skip Fridays. Extra credit 'twill probably haunt me, that lowly zero grade in BB350. It could be steric or esoteric, that carbons get so anomeric. I'm too hysteric, better let it go. This song's for BB350. Okay, great. Have a fun weekend, guys.
HPLCs in production plants, they don't set them up for
they-
amino acids, but you can set it up
right.
for any chemical substance.
Excuse me, yeah. And a lot of you, they do prep. So I'm
Yeah,
talking
yeah.
about an analytical sense here. And prep, we're talking ones can use giant columns and actually get a tremendous amount of stuff out of there. You bet.
Because I pulled it with the needle and pushed it in, and
Yep.
then it takes that and it powers it in. Just,
Right.
just like you were saying, I was amazed that.
It is amazing. You can generate, 6,000, 10,000 PSI in these things. I mean, they're amazing.
You got to actually be careful not to blow it up.
Oh, yeah. Oh, yeah. I did that one time.